Hello students, hope you understand the formation of male gametophyte in angiosperms. Now we are going to discuss the formation and development of female gametophyte. First of all, we'll discuss the pistil, megasporangium, that is ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. It has three parts: stigma, style, and ovary. The gynoecium, it may consist of a single pistil, which is called as monocarpillary condition, or it may have more than one pistil, which is called as multicarpillary condition. When there are more than one, it is called as, more than one gynoecium is there in a flower and all are fused together, the condition is called as syncarpus, or it may be free, that is called as apocarpus. Each pistil, each gynoecium, it has three parts. As I have already told you that it has stigma, style, and ovary. Stigma serves as a landing platform. It is the broad base on which the pollen grains they land after pollination, and style is a long tube, a cylinder-like structure which is present beneath the stigma, and the basal bulged part, which is somewhat oval in shape, it is called as ovary. Ovary consists of ovules inside it. It has several chambers or ovarian cavities inside, which are called as locules. Placenta is also located inside the ovarian cavity, and placentation, you all know very well that it is the arrangement of ovules within the ovary. Arising from the placenta, there are several megasporangia, commonly called as ovules. The number of ovules may vary in different flowers. It may be one in wheat, in paddy and mango, or it may be more in number, like in papaya, watermelon, and orchids. Let's discuss the structure of megasporangium, that is ovule. If we see the structure of anatropous ovule, it has a stalk called funicle. It has a hilum, a point which is the point of attachment of body of ovule with the funicle. Several layers or integuments are there, and a small pore called micropyle end or micropyle, which through which the pollen tube enters the embryo sac. And opposite to micropylar end is the chalazal end, which represents the basal part of the ovule. Enclosed within the integuments is a mass of cells, which is called as nucellus. The cells of nucellus, they may have abundant reserve food materials. Located in the nucellus is the embryo sac. It is the basic female gametophyte, and ovule, it generally has a single embryo sac formed from a megaspore through reduction division. Reduction division means meiotic division. So the process of formation of megaspores from megaspore mother cell is called as megasporogenesis. So when the ovules are formed, they are generally differentiate a single megaspore mother cell MMC in a micropylar region. As you can see in diagrams in figure number 2.8, it is very clear that how the formation of female gametophyte, how the development of a mature embryo sac takes place from a single cell. A large cell containing dense cytoplasm and a prominent nucleus is there. The megaspore mother cell undergoes meiotic division, and it leads to the formation of megaspores. So 4 megaspores are formed, but out of these 4, only one is functional. Other 3 degenerate and provide nourishment. The one which is act as a functional megaspore, further it undergoes several mitotic divisions. Nucleus divides by several mitotic divisions to and give rise to the formation of monosporic development is this type of development, and it leads to the formation of 7-celled 8-nucleated embryo sac. When the 7 cells and 8 nucleated embryo sac is formed, 3 cells they move towards the chalazal end, they are called as antipodals, and 3 cells move toward the micropylar end, which forms egg apparatus. Egg apparatus basically consists of one central egg and 2 synergids on the sides. At the center of the embryo sac, 2 polar nuclei are present, and later on they fuse to form a single cell, that is secondary nucleus, but it is diploid. So in this way, embryo sac is formed. This is called as 7-celled 8 nuclei because of the fusion of polar nuclei to form a secondary nucleus. This type of development is also called as monosporic development. Monosporic development means the development which takes place from a single functional megaspore. So I think the formation of female gametophyte is now clear to you. Just look at the diagram and the important points once. Next topic is pollination. As you have learned that male and female gametes in flowering plants are produced in pollen grain and embryo sac respectively. As both types of gametes they are non-motile, they cannot move, they have to be brought together for fertilization to occur. So this is done by the process called pollination. So pollination is defined as the process of transfer of male gamete of plants, that is pollen grains, from anther to stigma of same or different plants. This process of pollination, it is a mechanism to achieve this objective, because if the gametes are transferred, only then the fertilization can occur. So depending on the type of pollen grain, or depending upon the source of the pollen grain, the pollination is divided into three types: autogamy, patonogamy, and xenogamy. So first of all, we'll discuss autogamy. Autogamy, it is the process of pollination which occurs within the same flower. It is the pollination which occurs in the same flower, and it is rare in those flowers which open and expose the anthers and stigma. Some plants like Viola, Oxalis, and Commalina, they produce two types of flowers. One is chasmogamous flowers and other are the cleistogamous flowers. Chasmogamous flowers are the open flowers in which both self and cross pollination can occur. They have exposed anthers. But in cleistogamous flowers, only self pollination occurs, because they do not open at all, and to produce assured seed set, even in the absence of pollinators, they lead to self pollination and they do not open at all. So uh, these types of flowers, they are the examples of autogamy. Next is jatonogamy, which is the transfer of the pollen grains from anther to the stigma of different flowers of genetically similar plant. Next is xenogamy, or cross pollination, which is true cross pollination. That means it is in this process the transfer of the pollen grain takes place from anther to the stigma of genetically different plant. Now, both self and cross pollination, they have their advantages as well as disadvantages. First of all, we'll discuss the advantages and disadvantages of self pollination. Self pollination, as its advantages include the chances of pollination are more in this type of pollination. It maintains the purity of race and avoids mixing. It also needs not to produce a large number of pollen grains, because the pollen grains, they need not to be produced in larger numbers, because they have to fuse in the own flower only. Flowers need to possess devices uh, such as they have showy petals, presence of they need not to possess any scent or any nectar, etc., to attract the pollinators. Disadvantage of self-pollination is that the progeny continuously gets weaker after several generations, and there are less chances of production of new species and varieties. On the other hand, we are discussing the advantages and disadvantages of cross-pollination. Cross-pollination, it because it is the transfer of the pollen grains from anther to the stigma of genetically different plants. So its main advantage is that it leads to genetic recombinations and the production of new variations, and it leads to evolution also. Cross pollination, it results in healthy and stronger offsprings. Several crop plants such as mustard, sunflower, cucurbits, they give high yields if bees are available and cross pollination is allowed to occur. Variations caused due to cross pollination of disease resistant plant, it results in the production of seeds in self-sterile plants also. But disadvantage of cross-pollination is that it is not economical. The plants waste a lot of energy and food materials in unnecessary adaptations and devices to bring about the pollination. They have to attract the pollinators so that the pollen grains can, if they are, because they are non-motile, so they are taken to the female plant through several agencies. Cross pollination is uncertain because a factor of chance is always involved, and it involves addition of some undesirable characters or loss of some important characters sometimes.